we've covered glycolysis. I've talked about glycolysis. I've talked about how the equations of respiration, glucose plus oxygen goes to carbon dioxide and water, is essentially all a lie. Not really a lie. It is still true. It still bodes true. But the fact is, it's a lot more complicated and not as vague as one simple done and dusted chemical reaction. And while I've mentioned glycolysis, I haven't mentioned at all any of the digestions and metabolisms of fats and proteins. I have done the metabolism of alcohol in one of my videos. All of these end up turning into acetyl-CoA, one of the base molecules for all sort of human metabolism and digestion. But the sort of second main part of respiration is known as the Krebs cycle, and this begins with the formation of acetyl-CoA. But I covered glycolysis, and this ends up being a reaction where glucose is converted through several chemical reactions, enzymatic reactions, to end up turning into pyruvate. Now, pyruvate is an interesting molecule that has a few options for where it wants to go. It can turn into a few other proteins. It can go back into glucose. It can turn into oxaloacetate to go back into glucose. It can do so many things. But one thing it can do is turn into acetyl-CoA to essentially complete the reaction from glucose all the way to acetyl-CoA. And this happens in one simple done and dusted reaction where pyruvate turns into acetyl-CoA using the coenzymes NAD+ on the left side, NADH on the right side, also having like a CoA SH molecule that is the reason the CoA molecule can end up being added onto the acetyl group. You have CO2 that ends up also being excreted off the side of this reaction, and this reaction is catalyzed by the enzyme pyruvate dehydrogenase. Now you finally have acetyl-CoA being made from glucose, as I've talked about earlier. But how exactly does acetyl-CoA get fed into the Krebs cycle? The Krebs cycle, the cycle of sort of molecules, carbon chains, to end up converting back into oxaloacetate, which can then be combined with acetyl-CoA and then go round again to oxaloacetate and combined with acetyl-CoA. And that's essentially the process that I'm going to talk about in this video. I'm going to go through how all these, I think there's around 8 steps work, all the energy yields it produces, and maybe a few other interesting things that you may not have known about before. Now, this process, the Krebs cycle, occurs in the mitochondria of your cells. Once pyruvate has been made from glucose in the process of glycolysis in the cytosol of the cells, the pyruvate will end up moving and being transported to the mitochondria, where that sort of link reaction happens in a way between pyruvate and ends up going into acetyl-CoA. And the first step inside of the Krebs cycle is acetyl-CoA plus oxaloacetate ends up turning into citrate plus a separate coenzyme A molecule, and this reaction is catalyzed by the enzyme citrate synthase. There aren't any other coenzymes or any energy yields in this reaction. It's sort of like a boring and basic one in a way, but trust me, it does get more interesting as the process goes on. Now, the second step inside of the Krebs cycle is citrate turning into isocitrate plus water using the enzyme. The enzyme is the thing that catalyzes this reaction, aconitase. Now, I said that water is produced during this reaction, the reaction from citrate to isocitrate. Water is produced as a byproduct, and that is one of the ways the generic respiration equation worked. I said right at the start, the respiration, the thing that you may have been taught in school, is kind of all a lie. It's not a lie, it is correct, it's just way too vague for you to be able to fully grasp the concept of how this metabolic process works. Water is produced, and while the generic equation, glucose plus oxygen equals carbon dioxide plus water, that is one of the ways that water, one of the products of the generic reaction, actually is produced inside of the complete respiration process, and that is one of the ways that the reaction actually works. And there are a few other ways that happen inside of this process, as well as a few other processes that I've talked about in past videos and will do so in future videos as well. Now, the third step inside of this reaction process is isocitrate plus NAD+, which is a coenzyme, turns into NADH plus alpha keto glutarate, as well as a CO2 molecule. Now, this reaction is catalyzed by the enzyme isocitrate dehydrogenase. And like how I said before with water, ends up being produced as a byproduct, which is part of the generic respiration equation, CO2 and how this molecule is produced also applies to that sort of same thing that the water did before. And that's another interesting thing. That's another way that the equation bodes true. And I will probably cover, in case you haven't watched some of my previous videos, how the whole actual respiration equation does end up working out in the end through the processes like glycolysis, the Krebs cycle, and oxidative phosphorylation. But alpha-ketoglutarate, the main thing that's produced during this reaction, which is then used to actually produce the other Krebs cycle intermediates, is an interesting thing because I've never really gone through what an alpha-keto acid actually is before. Alpha-keto acids are things that are formed during the deamination of proteins and amino acids. Often, proteins and the things that make up proteins are amino acids. These often contain nitrogens, and nitrogens inside of the majority of the body's metabolic processes aren't really involved all that much, especially in the ones that I've covered so far. But when a protein enters the body through things like meats, through things like beans, and just pretty much every single food that you eat on a daily basis, these proteins have to have the nitrogens removed in some way, shape, or form to be involved in things like the Krebs cycle, as well as to be converted to acetyl-CoA, because obviously acetyl-CoA, and at least the acetyl part of acetyl-CoA, doesn't have any nitrogens. So alpha-keto acids are formed when you have to get rid of the nitrogens, because the nitrogen base, the amine base, ends up being replaced by a ketone. And the ketone is often next to the carboxylic acid group in these cases, and that ends up meaning that it becomes an alpha-keto acid. But another thing that's interesting about the deamination of proteins is that what happens to the nitrogen is a very interesting thing that is something that you also may have learned inside of school, and that is the fact that you can't exactly get rid of the nitrogens very easily. The removal of the nitrogens requires the production of ammonia. When you get rid of the nitrogen, you end up having to sort of let it be free on its own, and a sort of free nitrogen molecule that ends up having hydrogens saturated ends up being ammonia. NH3 is ammonia. Nitrogen can have a maximum of 3 hydrogens attached to it in the majority of cases, and like carbon can have 4 hydrogens attached to it. This all relates to its sort of chemical and electronic structure, which I may go through in other videos. But what's interesting is that NH3, it's a toxin, it's ammonia, it can't really be in the body, and so this ammonia, while it may not exactly be ammonia for an extended period of time whatsoever, is probably immediately converted after the deamination of those proteins into urea. And urea is a water-soluble chemical. It's also a toxin, but it's not as much of a toxin as ammonia is, and then this urea ends up being excreted when you go to the toilet. That is why your piss generally is kind of yellow, because urea is like a yellow molecule. So yeah, that's another interesting thing that you just simply may not have known before. But I did mention earlier that proteins can be made into acetyl-CoA, but they can also be made into straight up intermediates of the Krebs cycle, like alpha-ketoglutarate. And that's another interesting thing. There's a whole topic of sort of ketogenic, glucogenic amino acids that I will talk about in a video where I specifically just talk about the details of acetyl-CoA and all the wonderful things that it does in the body. But right now, I should probably get back to the Krebs cycle and all the other wonderful things that that does inside of your mitochondria. Now, the fourth step inside of the Krebs cycle is you take that alpha-ketoglutarate, you add it to an NAD molecule as well as a coenzyme A molecule, and this will end up turning into succinyl-CoA. It's quite a weird sort of thing to pronounce with a double consonant C. I may have got it wrong. I'll just put up what it spells like on the actual screen in case you don't quite understand, but that will turn into an NADH molecule as well as another CO2 molecule, again proving why the generic respiration equation kind of does work, but again is far too vague for you to actually get the full picture of the metabolic processes that occur inside of your body. And this reaction is catalyzed by the enzyme alpha-ketodehydrogenase. And the fifth step inside of this reaction is where you take that succinyl-CoA and you turn it into succinate. And the coenzymes involved are GDP as well as a PI molecule on the left side and a GTP molecule on the right side, as well as a coenzyme A molecule from where it sort of went on the succinyl-CoA molecule. And GTP are interesting things. It's GTP stands for guanine triphosphate, which adenine, just like adenosine triphosphate, guanine is like a nitrogenous base that is used inside of the production of DNA. GTP, I'm not as sort of aware about as things like ATP. I assume because GDP goes to GTP, it ends up maybe being converted into ATP, and that's why the Krebs cycle can have 1x ATP energy yield, or at least that's what I've sort of done and seen on some of the research that I've been doing. But some places said that it produced GTP, some places said that it produced ATP, so I assume one of them just may end up making the other into the other, or some kind of thing like that. Maybe I'll put a paragraph that would explain the full thing in a better way than I possibly can off the top of my head, but hopefully you kind of understand and get the sort of grasp of what I'm trying to say. And this reaction is catalyzed by the enzyme. I almost forgot the actual thing that catalyzes this reaction, which is succinyl-CoA synthetase. And the sixth step inside of this reaction is you take that succinate molecule, you add it to FAD, not sure what that stands for, but that will end up turning into fumarate plus FADH2 subscript. And what's interesting about FAD and FADH2 is FAD, if you look at the molecule on the screen, will end up having a riboflavin section on the FAD molecule. That is because riboflavin is needed to produce FAD, just like niacin is needed to produce NAD. That is why the B vitamins are such vital and crucial things for your body, because otherwise things like the Krebs cycle and a load of other chemical reactions simply will not work. But what's also interesting is that the B vitamins themselves are, like, as a class of vitamins, things that are known as energy producing vitamins. And you can see why they're energy producing vitamins, because they're used to produce certain coenzymes, or they are the coenzymes themselves that are used to produce energy, can be used in the oxidative phosphorylation electron transfer chain, which will end up yielding ATP, which can be used to make your muscles move and do a whole wide variety of very good things that you like. Now, the seventh step isn't as interesting as any of the reactions that I previously talked about. You end up taking fumarate, adding it to water, using the enzyme fumarase, and that will end up turning it into malate. A bit of a boring reaction, a bit of a basic reaction, but that's just how science and chemistry works, I guess. You need some interesting stuff, and you also need some pretty basic and boring stuff for you to get any joy out of the actually interesting stuff. I suppose maybe that's too philosophical or whatever, but I guess that's just how it works. And the eighth step inside of the Krebs cycle is you take that malate, you add it to an NAD molecule, that will end up turning into oxaloacetate plus an NADH molecule, and this reaction is catalyzed by the enzyme malate dehydrogenase. And that's it. You have oxaloacetate. Oxaloacetate plus acetyl-CoA equals citrate, using the enzyme citrate synthase, and boom, the cycle will continue and repeat, continue to produce energy-yielding molecules, and the total energy yield of the Krebs cycle, I will include the energy yields of the link reaction, which is simply just one NADH molecule, but then the Krebs cycle itself, you end up having 3 times NADH molecules, so that makes 4 times, including the link reaction, 1 times GTP molecule, which I assume ends up being used to produce actual ATP in another metabolic processes. I may be wrong on that, but I'm honestly not quite sure. But then you also have 1 FADH2 molecule, which, like NADH, can be used in the electron transport chain to yield a lot of ATP, which is a better high energy molecule for your body to have than FADH and NADH itself. In fact, just a quick disclaimer and aside that I've only just realised when looking at the bottom of my script, GTP doesn't actually end up being used to make ATP. It is only something that's used in the process of protein synthesis, which probably yields some sort of energy because it is needed. Protein synthesis is essentially the thing that produces a lot of enzymes and a lot of hormones, which I guess will yield energy in some way, shape, or form. I'm just not quite sure why on some bits of research I did, some said GTP, some said it produces ATP. Maybe, I'm not quite sure, I just chose GTP because I thought maybe it'd be a bit more of an interesting thing to talk about than the ATP that I've mentioned in a few of my past videos. And yeah, there was a few weird circle interesting diagrams that I couldn't quite wrap my head around, no matter how much I tried to understand it. But I hope I've explained this as well as possible so that you now understand the Krebs cycle as best as you possibly can, and now know a few more interesting things about how respiration works and how metabolic processes occur inside of the body. So with that, I hope you enjoyed the video. I appreciate it if you stayed this long, and I also recommend subscribing because I love this community of young tribal people who are willing to improve themselves so much more than you already have. And good luck.